Today, I will talk about our work, phylogenetic profiling of PAF1 complex associating proteins during development. RNA polymerase II associated factor 1 complex called PAF1C is a hepatometric protein complex, which consists of PAF1, CTL9, BO1, CDC73, and RTF1, and evolutionarily conserved. Additionally, WDL61 is present in some organisms, including humans. PAF1C regulates gene transcription by associating with RNA polymerase II, and PAF1C also regulates chromatin modeling through histone ubiquitination and maturation. It also has been shown that PAF1C is involved in cell proliferation, cell differentiation, and tumor suppression. However, the functional role of PAF1C in development has not fully clarified. Therefore, in this work, we attempt to identify evolutionarily conserved factors involved in PAF1C-mediated function in both humans and the simplest of molecular organisms, C. elegans. First, we constructed a dataset that interacts with PAF1C components in humans using BioGrid and UniProt. Second, we performed a phylogenetic analysis. In this analysis, the presence or absence of orthologous protein is shown as 1 or 0 in each species based on bit pattern. 1 is shown as black. The orthologs were retrieved with KEGG ortholog cluster. Phylogenetic profile represents evolutionary preservation, so the bit patterns have a high degree of similarity to each other and considered to be co-evolved and related. Eventually, we clustered phylogenetic tree based on similarity of bit patterns using a word method and calculated using the Euclidean metric. Third, human gene expression levels in each of the 37 tissues were obtained from the Human Protein Atlas, and C. elegans gene expression levels in each of the developmental stages were obtained from Expression Atlas. We built a database with retrieved C. elegans genome sequences, data from UniProt, and searched homologous proteins. These expression levels were normalized using Z-score for each protein and clustered using the similar method for the phylogenetic profiles. Finally, cellular component and biological process annotation was obtained using UniProt keyword. Next, the result. The dataset of 4,158 PAF1C components binding human proteins were constructed. The PAF1 component, CTR9, is conserved in eukaryote. WDR61, RTF1, and CDC73 were constructed from plenty. Leo1 and PAF1 were conserved across metazones. These results suggest that the largest molecule, CTR9, has been conserved for a long time, and other factors may be important for the acquisition of evolutionary diversity into animals. Next, by analyzing the difference and similarity in the RNA expression levels in each tissue, they were divided into 6 clusters, and the PAF1C components were divided into class 1 and 2. Additionally, we found that nuclear localization ratio of proteins belonging to class 1 and 2 were higher than that belonging to of class 3 for 5 and 6. These results imply that the expression levels of PAF1C components were co-regulated within each tissue. Next, focusing on the class 1 and 2 cluster protein of human RNA expression analysis, we reanalyzed the similarities in the developmental expression patterns in C. elegans development. RNA expression levels in C. elegans developmental stage were analyzed. and divided into 6 clusters. All components of PAF1C except CTR9 were localized in the same class. We defined 3 subclusters based on similarities in expression patterns, and we extracted the evolutionarily conserved subcluster genes as a result of phylogenetic analysis that were localized to the neighborhood of the PAF1C components. Finally, we characterized 8 PAF1C component-associated proteins and annotated as transcription. We found that most of these proteins interact with CTR9 and suggesting that they co-evolved and act as a component of the CTR9-mediated transcriptional machinery. Thus, we speculate that association between the PAF1C and these PAF1C binding proteins may co-evolve to act as a key molecule in the context-specific PAF1C function during development and co-evolution. In conclusion, gene cluster of the evolutionarily conserved PAF1C associated proteins were obtained using gene expression in human tissues and further analyzed during C. elegans development. Furthermore, to elucidate the evolutionarily conserved interaction between the PAF1C associated proteins and PAF1C, we would like to further analyze the functional relationship and network among genes in the same cluster. Thank you.